Welcome back to the fourth lecture on animal physiology. Today what we will explore, we will just talk about a reverse situation of positive feedback, which are very few which happens positive feedback in our body, but there is one example which I will give you. But detailed molecular mechanism, we will be talking about it when we will talk about the blood clotting. It is something to do with blood clotting, okay? And from there we will move on to the different sectionings of the body before we move on to the overall layout of chemical organization, tissue level organization and the cellular level of organization. Sorry, it should have been molecular level or chemical level organization, cellular level organization, followed by the tissue level of organization. Okay. So, coming to this class, which is, so this is our lecture 4. Here we will be talking about positive feedback loop. Positive feedback loop. And this positive feedback loop, what we will be dealing today, will be mostly related to the situation of blood clotting. So, say for example, I will tell you a situation. Say for example, there is a break in the blood vessels. So, let us think of the real life situation. What happens when there is a break in the blood vessel? You fell down or you have a scar, you see the blood oozes out from that place, right? Because the blood vessels gets ruptured, capillaries gets ruptured. Soon after that, what you see, after a while, the blood do not come out from that wound. Slowly the blood stops to come out, and eventually you see some form of a small semi solid kind of structure which is formed there, which we call as a clot. And over a period of time, as days passes by, that clot becomes harder and harder, and as all the tissues of the damaged location kind of repair itself, the clotted tissue, which has become already very hard, sloughed off from the layer, or it kind of falls out from there. So, this is what you see whenever there is a rupture of the blood vessel due to any injury, due to any accident, due to any scar or whatever. This whole process you can translate in terms of engineering in a simple way, as if there is a pipe which is carrying water and there is a leakage in it, and somebody has repaired the leakage. It is almost similar to that. So, in your body there are blood vessels which are carrying blood all over your body. For some XYZ impact, those blood vessels at some specific location got ruptured and it self heal itself. So, this process is called a clotting process, blood clots. But then this is what you observe. But now let us break the problem of what all events which takes place. Okay. So, let us put them in the way we put earlier. So, break in the blood vessel, this is the first event what you observe. Okay. Break or rupture, you can put it, break or rupture in blood vessel causing bleeding at a specific location in your body, okay, which leads to, so say for example, in a real life what is happening, these are the blood vessels which are supplying blood all over your body, and say for example there is some rupture which happens here. So, the blood started to ooze out from that location. Okay. So, this is the blood which is fallen. Okay. Now, as soon as the blood started oozing out from this location, so this part is the damaged tissue what we are talking about, okay, the this location, okay, which is your damaged tissue or damaged blood vessels, as well as the surrounding tissues. Surrounding tissues means all this location, these are all surrounding zones, okay, what I am drawing is in the orange color. So these damaged cells, they secrete certain specific chemicals. I am not getting into the details at this time. While we will talk about the blood clotting, I will tell you what are those chemicals. They fall under intrinsic and extrinsic process of clotting. These chemicals leads to the process of clotting to begin. Okay. Beginning of clotting process. So, as we are breaking down the problem, you see this one was step 1, this is step 2, and these kind of secretion of chemicals happen both from the damaged blood vessel as well as from the damaged tissue around it, because when you get an impact, say for example on your body, it is not only the blood vessels which gets ruptured, it is the muscle, it is the endothelial, it is the epithelial cell, it is the endothelial cells, they also get damaged. So these are the ones which secrete those XYZ chemicals, what you see out here, what has been highlighted out here in the picture. So these chemicals start the process of clotting, okay, which is the step 3, okay, followed by this clotting process initiate a positive feedback. It means this clotting demands for more clotting reagents to come into the place, additional chemical release. Okay. And this additional chemical release accelerates because the process of clotting, I am showing the acceleration with these 4 upward arrows. Clotting accelerates, and eventually this leads to the final goal, blood clot plug breaks in vessel wall and the bleeding stop. And this process, what is happening is here is a positive feedback. So, to summarize the process, you got an impact. At the impact zone, there is a rupture of the blood vessel as well as the damage of the surrounding tissue. So, now this vessel has to repair itself. The first reaction what takes place is the blood vessel, the damaged blood vessel as well as the surrounding tissue start secreting certain XYZ chemicals. These chemicals initiate the process of clotting and it sends a forward message, or in other words a positive support that you secrete more chemicals in order to completely seal the vessel where the rupture has happened, and this positive feedback leads to the sealing of the vessel, and final result is the bleeding stops, and followed by, as I was telling in the beginning, the surrounding tissue and everything repairs and we seal it and the body functions normally. So, now we have 2 examples, one of negative feedback in terms of thermoregulation, the other one is the positive feedback in terms of blood clotting. So, now I will recommend you people look for other positive feedback loops which are existing in our system, which it is almost like, think of it that there is, when you have to remember these kind of things or visualize, it is almost like at a zone there is a war zone, there is a small platoon or a small number or a small regiment which is fighting against it, and they ask for more reinforcement. You send us more reinforcement because we have injuries. Okay. Positive feedback exactly works like that. That you send us more and more reinforcement, we need to recover out because we are out of our resources. Okay. So, with this, I wish to highlight another very interesting aspect of physiology is that our whole body actually function on negative and positive feedback loops, and these loops are very important. Whenever you have to dissect out a situation, the first thing you should do, you should be able to draw them in like a block diagram, and you should be able to make the connectivity what all are happening, and at different point you will have different measuring technique to see whether this output is increasing over period of time or this output is decreasing over period of time. Say for example, if you look at this picture what just now I draw, how you can really quantify is it a positive feedback or a negative feedback. So, at every level, say for example, at this level you have to quantify. If you quantify with respect to WR2 with respect to time, you will see that these factors are all increasing. So, it means there is a positive feed forward loop which is functioning. Okay. So, you see in this simple diagram there are a lot of not only qualitative feature, but there are a lot of quantitative, qualitative as well as quantitative features involved in it. So, as a matter of fact, tomorrow's physiology is more of a quantitative physiology, because everybody wants to know that how much, say for example, I want to emulate the situation. So, again let us get back to the diagram. Say for example, there are say chemicals, these chemical release may be say A, B, C, D, E, just for your understanding sake. There are 5 different chemicals which are secreted. Now, I will be keen to know what will be the concentration of these chemicals which will be secreted at this specific zone. So, you know the damaged area. Say for example, I know x x square or x cube unit. In terms of vessel, if I have to go, I have to after the volume. In that volume, how much of these chemicals are released? So, in other way, what you are trying to figure out is the space. Out here is the space what we are talking about, and release of chemicals, second thing. So, if this is the space, damaged space, the types of chemicals, and the most important out here is this part with respect to time. So, you realize all the phenomena are function of space and time. So, when one can quantify a phenomena with respect to space and time, then one can say with certainty the limits of a system. Why this is important? You might ask the question other way. Why really we needed to find out these details or the fine prints? These fine prints are very important. Say for example, You have heard that soldiers get injured in the battlefields and they get hit by bullets or shells. So what happens in those situations? The first thing which happens, most of them suffer from heavy wounds. Heavy wound means there is a huge amount of blood vessel which gets ruptured. Say for example, out here, let's again come back to the picture for you to visualize. Say for example, here I showed a very small zone with a red circle. Now imagine in this blood vessel the damage is this big. This is the amount of damage which I am showing in green. Huge, say for example, a bullet injury or a huge shell hit out there, bullet injury or Now for this injury, now this is space constraints. Now what do you see in terms of damage to space, what we talked about, x cube. Now this becomes x plus x cube plus a huge area, okay, some y cube, and the chemical concentration what will be coming out, what you say, will also have to change, and not only that, time which will take. So the more bigger the injury site is, the more complex this equation, because if we know for this much unit area or this much unit volume, this is the amount which is released. Could we recalibrate it? What is the limit of the system? Is it repairable within that period of time that the person will not die? And what are our therapies? So there are people who work on several biomaterials who try to seal those big ruptures in the blood vessels, specifically for defense applications, specifically for those heavy injuries which takes place because of shells and all those kind of things. So you see a small problem, but you can think, you can stretch your imagination to a different level to see where it could all impact us. So this is what I wanted you people to think. Think beyond. Whenever I say think beyond, I mean I can teach certain things in the class, but that you have to think that will it be possible to make this repair there, or will this forward feedback loop or a negative feedback loop will act for a bigger area, because this is where comes from physiology to. So this is what you see whenever people talk about making patches, blood vessel patches and all these things, which is essentially, one second, let me write down, blood vessel patches. So this is what we are talking about, physiology to what I call it as engineering or material science, material engineering or bioengineering, whichever way you like to address yourself. It could be a bioengineering, it could be a material engineering or tissue engineering or biomaterials. So in order to understand any of these things, one has to have a clear idea how the physiology is taking its own course and what are the limits of a system. So as I told you in the very beginning, like these 4 courses I will try to kind of pinpoint certain things that where I expect you people to think much bigger, much broader. So these are some of those points where you should be able to think that where I can apply this, where this information is important, because we teach many things, but many a times we forget to make our students aware that where you really can apply it, because just imparting a piece of information is not sufficient. What are the implications of it? Okay. Next, what we will do after this, so as of now we talked about a system, okay, and let's say for example I say like this is the system I want to study and this is the anatomy of the system. How I am going to study it? What are the reference points? So today we will talk about all the reference points of the study, references of studying a body. Reference point. Fine, I haven't mentioned it. Fine. So, what are the reference points? So, say for example, or in other words you can call it a frame of reference for anatomical studies. Frame of reference for anatomical studies. So, say for example, you are standing sideways. Okay. So, this is your standing okay. So, this part, so for example, if you look at me, so I am standing like I am standing now sideways. Okay. Now, if you look at the sideways, you can see this picture. So, if you stand in a sideways, this is how it will look like. How we put the reference. This is the cranial part. Okay. The front of your body, this is called anterior or ventral part for body. The back of your body is called the posterior or the dorsal part, or the dorsal part, and the lower part which is called the caudal part of the body. Okay. Now, if the same person, instead of standing sideways, is standing with the front ways. So, for example, the hand, so if you are now in that situation, the upper part of your hands, so for example, this part of my hand, if you look at me, this is the part which is called the proximal end. Okay, you can see this. So, this is called the proximal end, and where you have the digits and everything, this is called the distal end. Okay. So, you will come across these words, and this part of the body is called the medial part. Similarly, the sidewise, which is the arms and everything, these are called the lateral part. So, these two are opposing each other, lateral and the medial. Medial is basically anything to do with the central part of the out here, whereas the lateral are on the sides. Okay. And okay. Now, similarly, lower part of the body, you have in terms of the legs, this is the proximal part P, and this one is the distal part. And there is two more words which are used. The upper part of the body is called the superior, and the lower part is called the inferior. So, the words which will you come across very commonly will be the proximal end, the distal end. So, always remember proximal is this part, distal is this part, central part of the body which is the medial part, and side which is the lateral part, lateral. Okay. Now, back of you is the posterior, front of you is the ventral or the anterior part. Okay. Remember this much, this is good enough. Okay. The cranial part and the caudal part. Okay. So, this is how you remember, and then we will come to the different planes by which you can do a sectioning of a system. So, for that we will move on to the next slide, the different kind of sectioning what you can follow. The sectionings, I will only talk about 3 different sections which are of major importance to you people: sagittal section, transverse plane, and frontal plane. So, say for example, what is sagittal section? So, sagittal section is essentially, say for example, I am standing here, okay. You make a cut like this. So, part of you make the nose into half, one half on this side, another half on this side. You make a straight cut like this on my body, right. So, that is called a sagittal section. So, half mouth on one side, half mouth other side, ear on this side, another ear on this side. Okay. You make the cut of the brains like this, like this. This is called a sagittal section. So, to draw it, it will be something like, say for example, so you are cutting the body like this. So, this is your nose in a sagittal section, part of the nose on this side, part of the nose on this side. If this is your lips, so it will be cut like this. Okay. So, this is called a sagittal plane. Next is the transverse plane. Transverse plane is that you are cutting the system like this. So, for example, this is the whole body and you are cutting it like this. Okay. So, that is called the transverse cuts, like this. In other words, if say for example, this is the cylinder and I give a transverse cut. So, these are the transverse cuts which are coming out. Okay. So, this is the transverse cut. Okay. Then comes a frontal cut. Frontal cut is very interesting. Frontal cut is something like, here you have cut it like this. So, in the frontal cut you are cutting this system in a, it's something like this. Hold on, your sagittal cut is coming like this, and when you are doing a frontal cut it will be something like this. You are cutting it like this. These are, so for you what is important is the sagittal cut and the transverse cut, and on the other side what you needed to remember is the proximal, distal, medial, lateral and ventral and the dorsal, and of course, the cranial, caudal, superior, inferior. So, these are few words of reference point which I wish you people to remember for rest of the course, because that is good enough. We do not need to go to the in depth detail of medical anatomy, because we do not need that for this course, but as long as you could remember the reference frames right, we are good to go. Okay? Thank you.